introduction down here. I'll just speak in here so that people can hear me. Well, thank you all for coming today. It's my pleasure to welcome Dr. Jason Hellman, who's an assistant professor of medicine and the associate director of the Flow Cytometry core in the Diabetes and Obesity Center. Jason received his BS in biology from UofL, and as an undergraduate, he was an author on 2 publications on aging and C. elegans. And then he did his Ph.D. in physiology and biophysics in Dr. Rooney Bhatnagar's lab. And during that time, he won first place in Research Louisville, basic science research competition. And he received the John M. Houchins Award for Outstanding Dissertation in 2011. And his Ph.D. work resulted in 2 first author papers plus 4 co-author papers. From September 2011 to July 2014, Jason was a postdoc in the Division of Cardiovascular Medicine, where he pursued his interest in obesity, inflammation, and vascular function, and he published 11 papers from that work. From August 2014 until April 2016, he was a postdoc in the Department of Anesthesiology, Perioperative and Pain Medicine, and the Center for Experimental Therapeutics and Reperfusion Injury at Brigham and Women's Hospital at Harvard. There he had 5 publications, including a first author paper in Nature Communication in 2016 on the Inflammation Resolution Program in atherosclerosis. Jason was recruited back to UofL in April 2016, and he's been very successful with 4 papers in high-impact journals, including a recent paper on dietary omega-3 fatty acids in modifying VO2 max and C-reactive protein in cardiovascular fitness, and I think what he's going to talk about is related to that. He's trained 1 postdoc and is currently mentoring 1 PhD student. He has served as a reviewer for the American Heart Association and for a variety of journals, and he's currently PI on 1 R01 grant, Exercise-Enhanced Resolution of Inflammation, and he's a co-investigator on 4 other R01s. So he's a busy guy, and I really appreciate Jason's taking the time to talk with us about his research today.
Thank you, Carrie, for that very kind introduction, and also for the invitation today to present and discuss some of our recent findings and some of the interests in our lab. So hopefully everyone can see the pointer here. Yes, okay. So today I'll talk about the role of lipid mediators in diet and exercise-induced inflammation resolution. And so we'll just jump right in. So the overview of the talk today, I'll start off by describing the role of innate immune cells and lipid mediators in the acute inflammatory response, as well as its resolution. Then I'll transition into talking about defective resolution of inflammation that we described in obesity-induced diabetes mouse models. And then I'll describe a little bit from postdoctoral work that was just mentioned, talking about lipid mediator imbalance in atherosclerosis. And then finally, some recent work that we've been doing here back at the university on my R01, looking at exercise-enhanced resolution of acute inflammation. And so I like to always start with, why is it important to study inflammation resolution? We've known since Hippocrates, to treat reduce fever and inflammation, simply, if one's willing, to chew on willow tree bark. But since then, we've got a little bit more advanced with the development of multiple pharmaceuticals, such as steroids, NSAIDs, COX-2 inhibitors, and now the more recent biologics. And while these have, clearly they have important roles in certain situations, there are also secondary effects that are, I guess, to say not optimized. So we know with prolonged steroid use, we have the development of osteoporosis, impaired wound healing, enhanced fungal infections. Of course, NSAIDs can give rise to stomach ulcers and bleeding, as well as compromised healing. I think we all pretty much are aware of the COX-2 inhibitors. They're wonderful in treating inflammation. However, were pulled off the market due to increased thrombosis and heart attack. And then more recently, the anti-cytokine or biologics are very effective in cases such as rheumatoid arthritis, but they do give rise to increased infection. because they dampen the immunity, right? They're immunosuppressive. And so in my lab, we're interested in understanding how inflammation resolves. And so if we think of a classic acute inflammatory response, upon tissue damage or microbial invasion, we have the formation of edema within seconds to minutes. And this is largely controlled by the lipid mediators that are prostaglandins, such as prostaglandin I2, prostaglandin E2, as well as the cystinoleukotries, which help to vasodilate the blood vessels to get more blood flow, recruitment of leukocytes into that area. And in doing this, this allows the infiltration of PMN or neutrophils into this infected area to help contain any potential microbes that are in the area. These PMN come in, they start to phagocytose and contain the microbes, and in doing so, they undergo apoptosis and die. And so they must be effectively cleared by monocyte macrophages that then come into the tissue to help clear away these dying neutrophils. And so you can imagine if this process is altered, it can throw off the duration and the magnitude of the inflammatory response. And so as I alluded to earlier, different lipid mediators have been shown to be critical regulators of this acute inflammatory response. Prostaglandins and leukotrienes are critical players in this inflammatory response at the early stages. However, a new class of lipids termed SPMs or specialized pro-resolving mediators have been discovered. They are namely the lipoxins, resolvins, protectins, and merisins. And these SPMs have been shown to be critical in this resolution or resolving phase. And they're critical because they prevent excessive neutrophil infiltration, but they also stimulate macrophage function and phagocytosis of these apoptotic PMN. And so this acute inflammatory response is nicely shown here. Let me see if I can switch back. You can see that the neutrophils rapidly infiltrate into the site of tissue damage, followed by the more slowly migrating green macrophages to help clear away these invading or swarming neutrophils. So, my lab is interested in understanding what controls the biosynthesis of SPMs, these pro-resolving lipids. So we know that the SPMs are largely derived from omega-3 fatty acids, EPA, DPA, and DHA. And through enzymatic conversion by 5-lipoxygenase or 15-lipoxygenase or 12-lipoxygenase, it gives rise to different classes of resolvins. So the resolvin E1 is part of the E-series resolvin family, and those are derived from the fatty acid EPA, whereas the D-series resolvins are derived from the omega-3 fatty acid DHA, as well as the production of protectins and merisins that are also derived from DHA. And so some of the signaling pathways and receptors for these SPMs have been identified on different cell types. We know that resolvin D1 exerts its pro-resolving function by binding to FPR2 receptor, which then has been shown to increase cyclic AMP signaling, to then drive increased efferocytosis and the formation of M2 polarization, which is an anti-inflammatory macrophage phenotype. Additionally, other resolvin D2 has been shown to bind to GPR18, again to drive increased phagocytosis and efferocytosis of apoptotic PMN. And so the discovery or the identification of these SPMs really challenged the previous concept that inflammation was just passively dissipated with time. Disappearance of local chemotactic stimuli and pro-inflammatory mediators just dissipated with time, and really brought forth a new concept that termination of inflammation is an active process, and that's rapidly turned on after the acute inflammatory challenge, and it's controlled or regulated by anti-inflammatory and pro-resolving mediators. And so a while back, we became interested in trying to understand if increased inflammatory mediators that are described in diabetic individuals, such as increased CRP levels, IL-6, and leukocyte blood counts. We were curious if these individuals with diabetes and this heightened inflammatory and presentation of heightened inflammatory mediators was due to a failure in resolution. And so we also knew from preclinical models that if we knocked out IKK or TLR4, junk TNF-alpha, that mice were protected against insulin-induced diet-induced insulin resistance, suggesting that there might be a role of this failure in resolution in terminating inflammation-induced diabetes. And so to test this, we use the obese diabetic mouse, the DB-DB, which you can appreciate is much larger, much more obese than your wild-type counterpart. And we simply gave these animals Resolvin D1, 2 micrograms per kg IP for 8 days. On day 5, we performed a glucose tolerance test. And we didn't really see a whole lot going on. What we found was that at day 5, glucose levels were pretty much the same between vehicle or RVD1 treated DB-DB mice. However, by day 9 of treatment, we started to see glucose levels starting to trend downward. And same with insulin, therefore our HOMA-IR score became significantly different. But we were really kind of unimpressed with these subtle changes. So we decided to extend it and treat the animals twice as long. And this time we treated for 16 days, whereby we performed a glucose tolerance test on day 10. And what we found was a significant difference in the DB-DB mice treated with Resolvin D1. They had improved glucose tolerance. They had a significantly decreased fasting blood glucose by day 10. And this was associated with an increase in circulating the insulin sensitizing adipokine, adiponectin, at both day 9 or day 17, suggesting there may be improvements in the adipose tissue that are maybe contributing to these improvements in glucose tolerance. And so we looked in the adipose tissue of these diabetic mice, and we quantified the formation of these crown-like structures. The crown-like structures basically are formed by infiltrating leukocytes into the adipose tissue as the adipocytes continue to hypertrophy and expand. And what we found is that Resolvin D1 treated animals had significantly less crown-like structures, suggesting that there was decreased inflammation in the adipose tissue. And so we quantified the presence of M2 or anti-inflammatory macrophages in the adipose tissue, and we found that Resolvin D1 treated animals had heightened levels of anti-inflammatory macrophages. And this was associated with a restoration of insulin sensitivity indicated by the phosphorylation of AKT following insulin treatment. And so this really was the first time, at least that we were aware of, that perhaps diabetes in mice may be due to a failure in the resolving pathways that turn off inflammation. But we were more interested in trying to quantify what those changes in resolution were. And so to do this, we employed a acute peritonitis model, which allows us to traffic leukocytes at different time points during an inflammatory response, but then also quantify the production of lipids at different stages of this inflammatory response. So this is a nice model. It's a self-limited model, meaning the animals will undergo an inflammatory response, but then resolve on their own within 72 hours. And this gives us the ability to really track changes in leukocyte populations within the peritoneal cavity over time and associate those changes with lipid mediators. And so to remind you again of what I showed a minute ago about the first infiltration of neutrophils followed by macrophage infiltration. And this is, as I was alluding to, this inflammatory phase at the beginning is dominated by prostaglandins and leukotrienes. We see those appear very early in this acute inflammatory response, followed late by the D series of resolvins to help resolve the inflammation. And so using this model, we can quantify how resolution changes by figuring out the time point at which 50% of the neutrophils are removed from the cavity. And so in doing this in wild-type and DB-DB diabetic mice, we found that the infiltration of the neutrophils reached a peak about 24 hours, but there was really no significant difference that peak inflammatory phase. However, about 48 hours, you can appreciate a significant difference between wild type and diabetic mice, suggesting that the removal of these neutrophils is delayed in the diabetic mice. And this was associated with an increase in Exin-5, or a marker of apoptosis, and that was despite any significant differences in macrophage numbers, suggesting that there was a defective clearance or removal of apoptotic PMN from the cavity in diabetic mice. So we tested that directly by isolating peritoneal macrophages from wild type and diabetic mice, and then feeding them FTSE-labeled Zymazan, little green dots that you can see on the left. And the diabetic macrophages were very defective in taking up or phagocytosing these Zymazan particles. Interestingly, however, if we treated these macrophages with low-dose resolvin D1, we could restore the phagocytic function of these diabetic macrophages. And so we were curious if this was unique to only the diabetic mouse, the DB-DB model in particular, or is this maybe a common phenomenon that may also happen with high-fat induced diabetes? And so we looked at low-fat and high-fat macrophages, and in fact, high-fat fed, macrophages isolated from high-fat fed mice showed defective phagocytosis. Additionally, if we take wild type and DB-DB plasma and treat naive raw macrophages, we can see a decrease in the phagocytic capacity after treating with a plasma isolated from diabetic mice, suggesting that it was a circulating factor that might be responsible for this defective phagocytosis. And so knowing that non-esterified fatty acids are elevated in both models, we decided to use a model fatty acid palmitate to see if that might be responsible for defective phagocytosis. And in fact, we found a decrease, or the percent inhibition of phagocytosis was dose-dependent with palmitate, suggesting that palmitate itself is decreasing the macrophage function. And so we started to probe a little deeper, and we looked at the expression of COX-2, and we found that it was actually being elevated in these macrophages treated with palmitate. And we see that the product of cyclooxygenase 2, prostaglandin E2, and D2 were both elevated following treatment of these macrophages with palmitate. Additionally, if we treat macrophages with these inflammatory lipid mediators, PGE2 and PGD2, we see a decrease in phagocytic capacity in macrophages. Interestingly, using the dexamethasone-induced thymocyte apoptosis model, we were able to demonstrate that DB-DB mice treated with dexamethasone had an accumulation of apoptotic PMN compared to wild-type vehicle treated. However, if we block EP receptors that are known to bind prostaglandin E2 and D2, we can reverse or decrease the diabetic accumulation of apoptotic thymocytes, suggesting that prostaglandins themselves are responsible for blocking macrophage phagocytosis. And so this allowed us to conclude that under normal conditions, we have the recruitment of neutrophils followed by recruitment of macrophages that effectively clear away apoptotic PMN. However, in diabetic models, we see that the defective phagocytosis and removal of these apoptotic PMN leads to chronic inflammation and the development of insulin, and presumably insulin resistance. And so around the same time, we were also reading papers, and we came across this paper in Investigative Dermatology, whereby diabetic skin wounds were shown to have this prolonged expression of GR1, which is a neutrophil marker, suggesting that perhaps even in diabetic skin wounds, we may see maybe a similar phenomenon where accumulation of neutrophils are persisting in the wounds. And so we developed a cutaneous skin injury model to test this idea. And so for this, we create two full thickness cutaneous wounds on the back of the mice. And then we place a silicone splint to keep the skin from retracting to more model human wound healing. And we were able to, or we do perform this, and we image the healing rate of the skin over 5 days to assess reepithelialization. And so doing this in wild type and DBDB mice, to no surprise, we found that diabetic mice had significantly delayed wound healing responses. But more interestingly, the intermediates that are known to drive the formation of SPMs were downregulated in the skin wounds of these DBDB mice. And so we added Resolvin D1 topically back to the diabetic wounds, and hopefully you can appreciate the enhanced healing that seems to be separating, at least statistically, around day 8. Additionally, we see enhanced granulation tissue formation in these topically treated diabetic wounds. And interestingly, we performed tunnel staining to assess the presence of apoptotic cells on the wound edge, and we see that RVD1 treated animals have fewer apoptotic cells present compared to vehicle treated diabetic animals. And that's quantified here below. And so we've now extended these ideas and concepts even in wild type or normal skin wounds. We see if we treat normal mice or non-diabetic mice with a Resolvin D1, D4, or D2, we see an enhanced wound healing capacity in these wounds. Interestingly, at least the effects of Resolvin D2 seem to be receptor mediated, as GPR18 knockout mice treated with Resolvin D2 show no enhancement in reepithelialization. Additionally, if we remove or knock out FPR2, which is a receptor for Resolvin D1, as I mentioned earlier, there's a defect basally in these animals in their capacity to reepithelialize. And so from there, we thought, well, perhaps this is a kind of the role of SPMs may be important in other chronic inflammatory diseases. And so we were interested in collaborating with our colleagues at Columbia, looking at the role of lipid mediators in atherosclerosis. And we know that atherosclerosis is a chronic inflammatory disease characterized by the infiltration of monocyte macrophages to help clear away lipoprotein particles within the formation of foam cells that then give rise to cytokines, MMPs, ultimately causing plaques to become unstable, or to form and then become unstable and rupture. And so we were able to get our hands on human enterectomy samples, and we isolated the stable plaque regions versus the vulnerable plaque regions, and we performed lipid mediator analysis on these different regions, and we were able to find that the production of Resolvin D1 was increased in the stable region compared to the vulnerable region of the plaque, suggesting that perhaps Resolvin D1 may be important in plaque stabilization. And so we performed a study in a preclinical model of atherosclerosis where we treated animals with Resolvin D1 or a vehicle control. In advanced lesions, we find that after RVD1 treatment that the lesions have enhanced Resolvin D1 compared to vehicle. But interestingly, we see a decrease in the pro-inflammatory lipid leukotriene B4. And that was associated with an increase in cap thickness following RVD1 treatment, a decrease in collagenase expression in the lesion, as well as MMP9, suggesting that RVD1 may be important, or at least a lipid mediator balance between pro-inflammatory and pro-resolving lipids may be important in plaque stabilization. And so these data have really kind of given rise to a new therapeutic avenue that's being pursued in atherosclerosis. There's been multiple other SPMs that have been treated or tested in preclinical models such as Merizum-1 or Resolvin D2, Resolvin E1. And as I mentioned, is really kind of driving a new therapeutic angle to study athero. But we were more interested in trying to understand, once the SPM production is depleted, are we just stuck adding it back? Or are there any endogenous mechanisms that may be important for restoring SPM biosynthesis? And so we became interested in exercise. We know that exercise obviously has anti-inflammatory effects. It's cardioprotective. While the anti-inflammatory effects are not well established, we do know that adrenaline or cortisol release in response to exercise leads to decreased TNF-alpha release from circulating monocytes. Additionally, IL-6 secretion from skeletal muscle has been shown to decrease TNF-alpha, as well as increase IL-1 receptor antagonist in monocytes and increase IL-10 production in macrophages. Really not surprising, in adipose tissue, following exercise, you have a decrease in the inflammatory macrophage content and a commitment increase in the anti-inflammatory macrophage content. And that's associated with decreases in pro-inflammatory cytokine production and increases in anti-inflammatory IL-10, as well as insulin sensitizing adipokine and adiponectin. And so we were curious if perhaps exercise may enhance SPM production and resolution, and maybe that's one of the anti-inflammatory effects of exercise. And so we subjected mice to 4 weeks of forced treadmill exercise. Following the 4 weeks, we performed our favorite peritonitis model to really assess leukocyte trafficking and quantify any potential changes in resolution. And so in performing these studies, what we found is that exercise mice actually display an enhanced recruitment of neutrophils at around the 12-hour time point compared to sedentary controls. However, the PMN are more rapidly removed in the exercise animals compared to sedentary controls. And there are significant differences in the amount of PMN in the cavity at 48 hours. And this is quantified as about approximately a 30% increase in the resolution. And so this is kind of an interesting phenomenon where we see increased recruitment, but then faster removal. And so we're still pursuing exactly, what this means, this heightened PMN recruitment. We're trying to figure out exactly what is driving that, and is it important. But nonetheless, in these studies, this enhanced removal of PMN was despite no change in macrophage numbers, again suggesting that perhaps macrophage function is enhanced following exercise. And in fact, we also see these are all wild-type animals. Yeah, just normal mice. Yeah. So following exercise, we see enhanced production of the SPM resolving D1, as well as the biosynthetic enzymes ALOX15 and ALOX5. So we wanted to test directly if macrophage phagocytosis is enhanced following exercise, and so we isolated peritoneal max from sedentary and exercise mice, and then fed them our FITSI-labeled zymazan, and we see increased uptake of FITSI-labeled zymazan in macrophages isolated from exercise mice. Additionally, we see increased resolving D1 production and no change in the resolving D1 receptor FPR2. And so it became what is the mechanism? How is exercise stimulating macrophage phagocytosis? And so knowing that exercise is associated with the increase in catecholamine production, we started measuring at least urinary levels of catecholamines following exercise, and we see no difference in norepinephrine. However, epinephrine levels were elevated in exercised animals compared to sedentary controls, as well as its immediate metabolite metanephrine was elevated. Additionally, white blood cells or leukocytes in the peritoneal cavity displayed increased expression of multiple adrenergic receptors that are known to bind the catecholamines, as well as the metabolizing enzymes COMT and MAL seem to be elevated following exercise, suggesting that perhaps adrenergic signaling might be important in exercise-induced resolution. And so we wanted to look at this more carefully. And so using bone marrow-derived macrophages in a dish, we treated them with increasing amounts of epinephrine, and we were able to at least show or test, find that resolvin D1 levels were elevated following epinephrine treatment. Additionally, we found the enzyme, biosynthetic enzyme for SPMs, 15-lipoxygenase, was elevated following epinephrine treatment, which was blocked when cells were pretreated with the alpha-1 antagonist prazosin, and not the beta-1 or beta-2 antagonists that were tested, ICI compound as well as pataxolol. So what these data suggested to us is that epinephrine, perhaps through alpha-1 signaling, may be important in stimulating resolvin D1 production and 15-lipoxygenase expression. And so that's exactly what we found in bone marrow-derived macrophages. If we treat with epinephrine, we see increased resolvin D1, which is blocked by the alpha-1 antagonist prazosin. Additionally, if we treat with an alpha-1 agonist to stimulate these cells in vitro, we see enhanced macrophage phagocytosis as well as the production of the SPM resolvin D1, which is blocked if we block the 15-lipoxygenase enzyme using ML351. And so we wanted to test this back in vivo. This was all in vitro data, so we wanted to test this in vivo. And so we moved back to our peritonitis model following exercise. We treated animals at both the 0 and the 12-hour time point with prazosin or vehicle. And again, we see this enhanced recruitment of PMN. However, if you focus on the solid blue line, you can see enhanced removal of the PMN compared to the sedentary controls. However, exercise animals treated with prazosin, the blue dashed line, we see a delay in the removal, consistent with the role of alpha-1 in exercise-enhanced resolution. And so we quantified or enumerated leukocytes in the cavity, and we find that exercise animals treated with prazosin have significantly more neutrophils, suggesting there's less removal. And that's despite no significant change in macrophage levels. Additionally, we find exercised animals have increased resolvin D1 in the peritoneal cavity, and this exercise-induced effect is blocked with pretreatment with prazosin. Additionally, we performed in vivo phagocytosis assay using latex bead, IgG-coated latex beads, and we find that prazosin treatment blocked exercise-induced phagocytosis. And so collectively, what we think is going on with exercise is that upon exercise and the release of epinephrine from the adrenal glands, the epinephrine can then act on alpha-1 adrenergic receptor on macrophages to increase the expression of 15-lipoxygenase and drive production of resolvin D1, which is then known to enhance phagocytic capacity of macrophages. And so we were really interested by these findings, and we were curious if this also persisted in animals fed high fat. What is the role of diet and exercise in enhancing resolution? And so to do this, we put mice on low-fat diet or normal chow or high-fat diet and for 6 weeks. And for 4 of those 6 weeks, we subjected the mice to our forced treadmill model. And you can see at the end of the 4 weeks of exercise, normal chow, low-fat, and high-fat fed animals show no significant differences between dietary groups in terms of the amount of work that they're able to perform following exercise, suggesting that the ability to adapt to this 4 weeks of exercise is not inhibited by diet alone. And interestingly, we see a slight, or it's still an increase in animals consuming high-fat diet following exercise, but no real differences in glucose tolerance or insulin tolerance tests. And this is only 6 weeks of high fat, so it's an acute model. There's no overt insulin resistance here. And so interestingly, what we find, however, is that normal chow animals have increased expression of M2 or anti-inflammatory macrophages in the adipose tissue and high-fat diet was significantly blocked that exercise-induced effect. Additionally, we see the expression of the SPM biosynthetic enzyme ALOX15 is increased in normal chow animals following exercise, and high-fat diet blocked that induction. Additionally, we find 4 SPMs are upregulated in normal chow exercised animals, and high-fat diet is blocking this exercise-induced production of SPMs in the adipose tissue. And so, the data here are very interesting to us, and we're trying to tease out exactly, what is responsible for the induction of the SPMs, which cell type. Is it the adipocyte? Is it something in the SVF fraction? And so we isolated adipocytes from the SVF, and we looked for biosynthetic enzymes of SPMs, ALOX15, ALOX5, as well as the metabolizing enzyme HPGD, and we see no significant increase in the adipocyte fraction. However, in the SVF fraction, we see elevated ALOX15 expression in normal chow animals following exercise, suggesting some cell type present in the SVF fraction may be responsible for our increase in whole tissue measurements. And so we isolated or sorted F480 positive cells from F480 negative cells. The F480 is a marker of macrophages, to pull out the macrophage population from the SVF. Following this, we get about 80% enrichment in macrophages, and we see that, in fact, ALOX15 expression following exercise is being induced in the F480 positive fraction and not in the F480 negative fraction. And this effect is blocked by high-fat diet. So what we think is that following exercise, we're seeing an increase in the expression of ALOX15, specifically in adipose tissue macrophages, which is being blocked by high-fat diet feeding. And so, given our data showing the importance of epinephrine, at least in our acute inflammatory model, we looked at the adrenal glands grossly in these animals following high-fat diet. Really no significant differences popped out to us by looking at them just by gross histologically. However, we performed PCR for biosynthetic enzymes that produce epinephrine, and we see that PNMT, phenylethylamine and methyltransferase, was significantly downregulated both at the mRNA level as well as at the protein level by high-fat diet. Additionally, we see epinephrine levels following exercise are decreased with high-fat feeding. And so we think that high-fat diet is blocking epinephrine release, and then this is preventing the pro-resolving effect of exercise, at least in the adipose tissue. And we think upregulation of SPMs may be important, not only for stimulating macrophage function, but also because macrophage function is tightly regulated by intermediary metabolism. We know inflammatory macrophages have a break in the TCA cycle and accumulate succinate to help drive pro-inflammatory cytokine production, whereas anti-inflammatory macrophages have an intact TCA cycle to drive ATP production. And we see if we treat at least bone marrow-derived macrophages with SPMs, we can increase mitochondrial respiration. So we're currently probing pathways to try to understand exactly how SPMs may be altering macrophage function, potentially through regulating cellular metabolism. And so in summary, obesity-induced insulin resistance is associated with delayed resolution of inflammation and dysfunctional macrophage phagocytosis. And stimulating resolution with resolving D1 treatment improved insulin sensitivity and decreased fasting blood glucose levels. Additionally, apoptotic cell clearance and diabetic wound healing is stimulated by topical RVD1 treatment. And then I also shared with you some data involved in atherosclerosis and plaque stability, which is modulated by lesional lipid mediator content. And some of our more recent data, the exercise-enhanced macrophage phagocytosis and resolution of inflammation is dependent on adrenergic stimulation. And so, we think that perhaps the prevailing idea that exercise is an anti-inflammatory event, it may actually be that exercise is a pro-resolving. And so you're not just blocking your immune system, but perhaps you're actually stimulating your immune system to do its job more efficiently. And we think we have some data that this may also persist in humans. And so looking at the NHANES cohort, 435 individuals with a mean age of 33 years old and a BMI of 27.8. We see that the association between VO2 max and CRP is obviously inversely related, and this has been shown many times. However, interestingly, it seems that the levels of omega-3 fatty acids may be modifiers of this inverse relationship, and in particular, the alpha-linoleic acid, as well as docosahexaenoic acid, DHA, which is a precursor for D-series resolvins, protectins, and merisins, seem to be important effect modifiers for this inverse relationship between cardiovascular fitness and inflammation. So with that, I'd like to thank people in the lab, Jenny, Ernesto, for doing the majority of the work, as well as Will Lynch, a former master's student in the lab, and Dan Riggs, who has done a lot of work looking at the role of fatty acids and this relationship between CRP and VO2 max, as well as collaborators in my previous lab, Matt Spite and Brian Sansbury, who's now at the university, and their help in doing the LCM, SMS, and adipose tissue following high-fat diet and exercise. And then, of course, the funding agencies. And then today, this is the event code that I should display. So with that, I'll take any questions. Yes?
The GPCRs that are the receptors for the resolvins,
Yes.
Are they expressed only in macrophages or are they elsewhere?
No, so in the wound healing experiment, we found that keratinocytes are actually, when we treat them with SPMs, it stimulates migration of keratinocytes, and we think that's responsible for the enhanced reepithelialization. So they're not just present on immune cells, but they're also present on endothelial cells and smooth muscle cells. And so a lot of work is underway now trying to figure out what are the other non-immune related effects of SPM signaling.
So when you do this, you draw the sample immediately after exercise, or
this is 24 hours after their last bout.
after their last bout?
Right.
So the epinephrine elevations
Before that. That's right. So the epinephrine effect is immediately following exercise. That's
okay. So the epinephrine effect is immediate, but the resolvins and other things you're seeing.
correct. So that was one of the reasons why we had to go to an in vitro model to really see if the two events are related. But that's a good point. We haven't really teased that out in vivo, other than using prazosin to block alpha 1, showing actual epinephrine binding. That's a good point. Yeah, we should probably address that a little bit more carefully. And there, the cycling of these receptors, adrenergic receptors, may also play a role in their signaling. So a lot of newer work's coming out that alpha 1 cycling into the nucleus actually has a different signaling role than membrane signaling. So there's a lot there to understand. But
And the injury, for the peritonitis, it's a single dose, correct?
Correct.
So whatever is the irritant is gone after that?
That's right. Yeah. No, the neutrophils, there's none in the cavity, and then after this low dose Zymazan, which is like a yeast cell wall membrane extract, it's pretty much gone within 12 hours. The neutrophils, 20, 30 million neutrophils that come into the cavity, and they remove it.
Because one of the problems that we have in trying to extrapolate these into clinical situations is the fact that whatever is irritant, if I have a wound infection, that thing is going to be there much longer. So if there's continuing inflammation, driver of inflammation, you still need that inflammation.
That's right.
What do you have to do?
That's and so that's kind of one of the ideas, right? Is that perhaps the inflammatory response is not robust enough, and so that's why I'm actually kind of really particularly interested in this exercise enhanced recruitment of PMN, and it might be that we need to hit some threshold of inflammation, not only to start removing, say, certain bugs or whatever the inflammatory stimuli are, but then to also kick off the resolution phase. And so if you can't reach that threshold, then you have this smoldering inflammation that just persists. But yeah, that's a good point. And what is blocking that? I'm not exactly sure. Yeah.
And the other question is, this is aerobic exercise.
Correct.
It's running.
Correct.
So one of the questions I have is, does resistance training do it?
Yeah.
I mean, because one of the problems with people who have wound infection, like which is mostly in the foot, is they cannot run.
Right. Right.
Don't tell them about no weight there.
Right.
So can they do,
resistance exercise
training and will it help? Because we had done a study looking at resistance training in elderly diabetics. In human studies, it's the data on what actually happens to inflammatory markers after exercise is very complex. Depends on what you study.
Correct.
And which particular marker you're going to look at. How long after exercise and things like that. And it was there. There was a lot of changes, but there's no consistent effect.
Right. Yeah. Right.
So that's why I was wondering, whether other types of exercise.
So with aerobic exercise, the release of catecholamines is intensity dependent, right? So you have to exercise like 60-70% max capacity in order to drive catecholamine release. I'm not sure, and if catecholamines, if what we found in the mouse translates into humans, I'm not sure catecholamines are going to be stimulated to be released with resistance exercise.
And if you said the dissolvents you're seeing 24 hours later, so if you do short bursts of exercise twice,
right?
Enough?
Is that
will
enough?
It increase the dissolvent
Right.
later, or will it just be?
Right. Yeah. We don't know.
Because it may be difficult to do bursts of exercise. I mean, long period of exercise, but try to 5 minutes in the morning and 5 minutes in the evening, like again.
Right. The high intensity interval training, right? So to kind of go out and run as much as you possibly can for 10 minutes and then be finished. Yeah, we don't know. We don't know. Thank you.
Stop sharing. You'll have some questions.
Stop sharing. Okay.
Should be able to